Well, how are you?
I'm doing great, thank you. Hey, I'd like to compliment you guys. So far, this program's gone very well. I'm really impressed. Congratulations.
Well, thanks. I appreciate you being here. So, if you wouldn't mind, as we've done customarily, give us a little bit of background specifically for the audience so that they understand where you're coming from, where your expertise is, and then we'll jump into your case study, which will bring us to the end of the program prior to the afternoon networking session.
Great. Yeah. Well, I've been in the as all of our speakers today. I've been in the industry for a long time, about 30 years. I've worked for Thermo and most recently Cytiva before moving to DPS Group, a facility design and build company. And I've spent my recent time, I spent my entire career developing products and processes for biomanufacturing. For the most part, back then it was MABs and enzymes and media development. But recently I've been working in some of the newer technologies that are related to the world of biopharmaceuticals and biomanufacturing that is expanding tremendously. I've spent quite a bit of time in 3D cell culture, including 3 and 4D bioprinting. I've been working in exosome manufacturing and publishing reviews on the manufacturing of extracellular vesicles for as carriers and as therapeutics. I've been interested in Industry 4.0 and just recently published a new paper on ATMPs and artificial intelligence. So artificial intelligence and machine learning is an interest of mine. And also then sustainability. And this is not biomanufacturing specific, but it's one of the now growing concerns and design criteria in processes and equipment, the sustainability of the process. And in that regard, I've been working with the BPSA, the Bioprocess Systems Alliance, looking at the increasing the sustainability or reducing the environmental burden of single-use manufacturing. And I've been working with Xavier University and their artificial intelligence and operations group, looking at artificial intelligence supporting operations in biopharmaceuticals.
Excellent. Sounds like you're the right man for the job.
Well, I'm spread a little too thin maybe, but I've been interested in a few different areas.
It's good. You know, the old adage, busy people get the most done.
Yeah.
I guess this is, as I said, case study number 6 out of 6, and we've got about 180 years of cumulative expertise in the case studies today. So it's like starting back. You mentioned Industry 4.0. It's like if we went back to Industry 1.0 and it's 1841 and we run until now and encapsulate all that experience in one place.
Well, it's funny. A lot of that older experience, it's amazing how different things were back then for most of the speakers today. It'd be fun to just sit around and chat about what the industry was like when we first started.
Right.
Quite amazing how different it was.
It is amazing. And so let's get to your case study in particular, the challenges of, I don't want to spoil the surprise for you. Why don't you launch in and tell us about the background of your case study?
Well, great. If we go to the next slide, I've got a little outline of introduction. In the title, I said that I'm talking about ATMPs, Advanced Therapy Medical Products. And the interesting point about that is that as we talked about when I started, we were making MABs and enzymes for the most part. And the contrast now is there are so many different entities, so many different types of products. It's unbelievable. Right. Sometimes we get a little focused on our particular product and aren't keeping up. But if you keep up with the diversity of entities and platforms, it's just hard to imagine. So we say ATMPs. ATMPs cover quite a broad spectrum of these products, mostly in the US that we refer to it as cell and gene therapy. But the term technically excludes certain products and includes a broad range of them. But even non-ATMP modern products are highly related in many aspects. So one problem we have in talking about intensifying the process is that we have both prokaryotic and eukaryotic culture. We've got animal cells, including human cells, to grow. We use viruses, oligos. We've got bioreactors and fermenters and chemical reactors. We've got viral vectors, non-viral vectors now. But there are some common themes, for example, for today that I can lean on to talk about intensifying the process. So with that, what do we mean by intensifying the process? And there are actually a few different definitions of intensifying the process. I liked what Sartorius came up with, and I've got the reference for that, the link below. But they refer to it as a holistic framework to maximize overall productivity of the unit operation, the manufacturing process, or facility output. And I like that. And that's the way I look at it. And the difference is that some people, for example, will include product quality. They'll say that improving product quality it could be a process intensification. I don't like that, because ultimately, if you include every improvement as a process intensification, then any activity in the process is process intensification, and it loses its meaning. So I like the idea of this definition here Sartorius came up with.
Yeah, and I guess it makes sense to start at the front end of that too, and say everybody would agree, well, we should undergo process intensification. That's like, asking the question, do you want to improve a process? You say, well, of course I do. And ultimately that boils down to, I guess, the idea of the market demand. So there's a demand for advanced therapies, and when the market asks, we should be able to deliver. So if there's a demand for greater ATMPs, you want to be able to react. So that brings us kind of the burning platform of why do it? And the answer is because it's the right thing to do, because there are patients and consumer elasticity of demand that says we need this.
That's a good point, Ben. And so there's two related points on that. One is that people have always been intensifying the process. And we used to make milligrams of product per liter when I started, and now we're making multiple grams. And that's an intensification. It's always been going on. I think that the term comes up now because we have so many more tools. And so we're now kind of giving a label to all of these tools that we can use to intensify it, and approaches. And then the second one is, yes, for ATMPs, getting enough in some particular products, it's a big challenge to get enough product out. And we'll talk more about that in the upcoming.
Great. Yeah, so let's talk about it. You've got a challenge, which is to identify the current and next generation technologies to do this process intensification. So what options would you say currently exist from which you need to improve things?
Well, if we move to the next slide, I first listed out the gaps or the needs, and if we could run through that quickly.
Yeah.
And well, okay. So first of all, with ATMPs, there are separate categories of needs right now. There's particular challenges in their scale up and their scale out, and some of that was introduced earlier today. There's a lack of experience in a lot of processes. As a matter of fact, some of these processes are absolutely unique, and if the product hasn't been launched yet, there isn't any experience. And there's a lack of best practices, and this is an issue I'm trying to make more. How do you improve something when you're the first person doing it? And then there's issues with cells as a product. It's a different process trained to consider the fact that you can't terminally sterilize, and you've got to maintain sterility throughout. And as was brought up by I think Morton Monk, there's also the issue with the diversity of molecular forms of the product, and this exists in a lot of our vectors where. And this isn't even very well known by people who aren't specialists in the area, but not only are there full and empty viral capsids, but there are incompletely filled capsids, and there is a diversity in the structure of the capsid. There is a diversity in the nature of the protein components of the capsids and the way they assemble. And someone has described that for one vector, there was as many as 50 different forms of the viral capsid. So there's right now an issue with the fact that when we analyze a batch of, for example, viral vector, in some assays you get a complete average, full and empty, partially full, different associations of the capsid proteins, and you get an average. And sometimes the average is helpful, but oftentimes it's not. You really want a point-to-point analysis. If you've got a certain value, is that every capsid has that value, or is it that half have nothing and the other half have twice? That type of thing. But we'll talk a little bit today about how we have tools now to actually discover the point-to-point distribution of the quality of these products. So that's some of the individual gaps that that exist in general with ATMPs. And then I've broken this up into pots of areas of demand for process development, for manufacturing, and for the facilities. And all three of these can be worked on to increase, to intensify the process. Now, looking at the process development, one issue there is that we're getting data from many different sources. We're getting it from the clinic, we're getting it from development activities, we're getting it from other related operations. There are times when, for example, large-scale cell culture, where we already have some knowledge, we can apply it to the current cell culture. Another issue is the siloed data. A lot of analytical equipment or laboratory results exist separately in isolation, and there's a big drive to coordinate them, to integrate the storage and analysis of them. There is often a lack of real-time or near real-time data. Offline analysis that takes hours or days is a real slowdown in process development. There's a limitation in the understanding of the cell biology and the omics of the process. We're looking at the immediate values of performance and of culture and of cell state, but there is often a lack of understanding of the molecular biology and cell biology behind these different results. And we don't, in trying to develop processes and modeling, we often don't have enough information to truly, the process control, we don't have enough information to develop good mechanistic models. So we're kind of guessing, and it's a difficulty in setting up a design space if you're relying only on the practical, the existential results of a reactor, rather than understanding some of the background to help box in a multi-dimensional design space of where you want to operate within.
Right.
And then in process control, there are a few different, let me just, because we want to move ahead, but one is that as we introduce artificial intelligence and machine learning and machine vision, things like this into the processes, there's a concern about the regulatory aspects of how we're going to handle that type of conclusion or that type of information. So that's the process development. Then there's manufacturing issues. Just to, I don't know how we're doing on time here, but there's large-scale stem cell culture, large-scale primary cell culture for allogeneic approaches, and then there are the scale-out issues that we've already kind of introduced for autologous, where we've got multiple small-scale cultures and how to operate them, coordinate them, and handle each individual cell line. Every individual sample in an autologous is a different cell line. It's going to behave differently, and it's difficult to set up a consistent process to handle such divergently operating and performing cultures.
Yeah, let me pause you there on the autologous end. So we got a question that was solicited prior to the event, and that is, will any part of autologous cell therapy manufacture copy the bioprocess intensification route, or is selecting and modifying the right cells so key that intensifying culture or reducing workspace becomes nearly immaterial?
Oh, well, no, it doesn't become immaterial, but there is a difference. If you're making a protein biological and you're working in CHO, well, there you've got a real repertoire of information about how that CHO line behaves in generic form or in your particular platform, K or some particular strain. if not past experience with the exact line or the exact product if you're doing a follow-on, for example. But here, you get a patient sample that's never been cultured before, and it's going to have a unique performance, and it has unique demands that'll respond uniquely to your process conditions and your operating procedure. Now, some of these uniquenesses are infinitesimal and not measurable or not important, but often they are, including failure. And the fact that each patient sample is critical for the therapy, unlike a batch of CHO cells, if the batch fails, you can make another batch and you got the same product. But there are times that an individual patient's culture, it's a one-time deal. You have one shot at saving this person's life. And so being able to optimize the ability to handle these diverse cell cultures is important for not just throughput and footprint or volumetric productivity, but for patient for safety and the importance of the criteria of the final product, which doesn't exist for a map.
Right. So given the list of gaps in the current state, what technologies do you find represent the technology trends that are worth selecting? Which ones have you seen be the most effective?
I'll go to that, but I'd like to set up, I've got a little bit of a case study I'm going to kind of talk about, and that is DPS Group. I'll just set up kind of the gap on this. I'm skipping through a lot of what we could talk about, but I want to introduce this, that DPS was given a challenge where they had a multi-floor, 3-floor facility that was kind of fallow at this point. And the client wanted to develop a recombinant protein and a viral vector manufacturing facility, and had some issues like the utilities were common to all the floors. And so I'll talk as we get up, as we move forward, I'll finish up with what DPS did to optimize this facility for efficient manufacturing of this protein and vector. Okay, so then you're asking about what tools we have to optimize. Well, as I introduced, we want to know more about the metabolism of the cells and the omics involved in the type of cell you're culturing. If it's a peripheral blood, a white blood cell, killer cells or T cells, or if it's a tumor cell, and to know more about the background, the basic idea of their metabolism and their needs and their cell surface markers. And that's unbelievable. It's just the amount of knowledge we're gaining about this is increasing dramatically. It's if you don't keep up with it, we can't even review it today. But such techniques as organ on a chip will allow a very rapid screening of processes and media and equipment to produce an efficient culturing process for an organ tissue, for example, because these organ on a chips give you thousands of experimental points in a truly biomimetic fashion. You've got more than one cell from a particular tissue that's generating the activity of that tissue. It's not just a cell line. It's not just a T25 flask of monolayer of one cell from a liver, but you've got multiple cells interacting, giving you liver function, which allows you to really test, for example, culture conditions on what this tissue, how it would respond, or how the final product will affect the tissue. It was mentioned that there's a tremendous growth in high throughput screening and automation in process development, and it's giving us many more data points, more advanced DOE. We've got much better statistical and even artificial intelligence driven design of experiment type of development that give us more efficient experiments to optimize in. We've got tremendous advances in probes. There are many more technologies in developing probes to give us immediate readings on existing tests. And we're moving from more siloed to network data, more multivariate data processing, rather than individual scientists looking at the results from separate assays and mentally integrating them or hypothesizing. We're getting true multivariate analysis and the ability to more accurately combine the results of some of these analysis and probe readings. And there's even now with artificial intelligence driven a curation of the databases that do exist or that are being generated from older equipment. And there's even now, there is a vendor of an AI-driven algorithm that will convert relational databases, which is what a lot of equipment is turning out, to databases that are more amenable to artificial intelligence manipulation and coordination and analysis.
Yeah, I mean, it's a beautiful thing. I think with all the advances in the technology and the database approaches, it becomes, almost like we talked about earlier. We've got Industry 4.0. There was 3.0, sort of 2.0, but every time one of those iterations happens, it's like rebooting the matrix, and things don't scale linearly. And so this is like yet the next shift where all of a sudden there's the seismic shift from where we were to where we'll be next. It's not just continuing along on this straight linear path.
And one of the problems is that it's happening in so many areas. In our probes, we've got so many different surface plasma resonance, piezoelectric, thermally based, Raman, near IR. There are so many different just technological approaches to monitoring the reaction or the culture, and then there's even now AI-driven observability analyzers that help look at with these mountains of data that we're generating with these new probes and analytics. There are these observability analyzers that are helping to discover early on in model building what data is valuable, what's contributing to the analysis, what's redundant or not valuable or doesn't change or isn't related to the consequences we're trying to measure, the process parameters or quality attributes we're interested in, and it can help weed out and filter down and reduce the data that we are then left with to use. So from the probes themselves, from the analytics, the omics understandings, and then the algorithms themselves and the data processing and the results generation, the model building and the model training, there are rapid advances month by month in all of these areas.
Yeah, indeed. So let's take some of that characteristic and bring it into where you go next with your case study. So take us home. Where do we go from the gaps in the current state?
Well, okay. So then, if you're looking at, for example, the data that's coming in, we've got, for example, clinical data that comes into both process development and manufacturing. It's difficult to handle. It's often poorly labeled or inconsistently labeled. This is from hospital to hospital or from geographical region to geographical region, and we're developing AI-driven data curation that will handle this, giving synonyms to the way values are identified. If you talk about a person has a high temperature or a fever or a temperature of 39 degrees, or there's different ways we can describe the same thing, and these algorithms can help with that inconsistently labeled data, or even deal with unlabeled data. And they also help in the areas of security. We've got HIPAA issues and we've got data security issues, and these modern algorithms are helping us with that. Let's see. So for expansion, this is in development and early manufacturing. For a lot of these entities, we've got an issue with the fact that there is zero market for this exact entity at this point. I mean,
Right.
there's need, but it hasn't started. So you're looking at 6 months or 6 years of lower level production, but you're looking forward to a large scale manufacturing, and you've got a concept of where the market's going to go to, and how to efficiently design this so that you've got a facility that will efficiently operate early on, and then have this expandability. And that's one of the facility issues that DPS is looking at. But it also occurs in just the process design. One interesting thing going on is in the automation of autologous cultures. There's the big issue of tech transfer from operating in T flasks in an incubator to working in a manufacturing environment, and that tech transfer. Everybody wants automation, and there are, as everyone knows, there are individual semi-automated and automated processes for particular operations in an autologous cell culture, for example, and gene therapy. But there are limitations. One of the limitations is that there are still open steps. We're getting packages shipped from hospitals that have to be handled as a contaminated material. We want to transfer that to an area to handle it sterilely and transfer that sterile contents to this automated piece of equipment. And then at that point, as I mentioned, one of the big issues here with cell and gene and many gene therapies is that there isn't any ability to sterilize after that point. So a contamination is you don't have to worry about endotoxin because you can't have any prokaryotic culture. So that's an issue in the process, but there's tremendous advances. People have heard about Mateni's work and the cocoon and these automated instrumentation coming along the line, but there's also some newer advanced thoughts. With some of these instruments, the cell handling, the culture itself is in its control system, the computer, the automation, it's all part of one package. So each sample has its own equipment to handle every step of the process. But there's a couple of offerings now of automated equipment that has a cassette-based handling. So you can punch in a sterile unit to culture the cells, take that out, put it in an incubator, it's sealed, it's closed, put that in an incubator, and then start another culture in the same process control and automation equipment, thereby saving expense. Whereas if you have 10 of the older style automation, you have to buy 10 complete automated units to process 10 samples. But with a cassette-based system, you can buy one and then pump in 10 different cassettes and have them culturing in an incubator. So these advances, as I said, it's just hard to even catalog all the different growing, real commercialized technologies that help us increase the process capability, the volume, whether it's volumetric productivity, whether it's footprint productivity, whether it's cell-specific productivity, or in vector manufacturing, volumetric productivity. All of these different technologies we've been touching on today will help us to increase it dramatically in some cases.
Mm-hmm. Yeah, and there was one other question that had come in, which it might be too general to ask here, but what the heck, we'll ask it anyway. It's the holidays. What is the cost of it?
Well, that's interesting. With respect to cost and sustainability, what I'm really excited about is talking about the serendipity or the win-win approach to cost of, for example, automation or automation and sustainability with product intensification. Because we often think, well, gee, automation is very expensive. But if you look at the performance, you look at the failure rate, you look at the cost of personnel, in the big picture, automation can be much cheaper, even though there's an initial output for investment capital. expense. And then, and similarly in serendipity, in sustainability, people think, well, to do something in a more green way is going to be much more expensive. And yes, I care about the world, but I'm launching a new product and I've got to be profitable. Often, process intensification directly relates to a greener approach, a less environmental burden, a more sustainable process. For example, we were hearing today about reducing N-1 or N-2 steps and intensifying the process train. If you can, through high density seed or high volume seed into a reactor, if you could skip an N-1 reactor step and go directly from seed to an initial culture to a final reactor, say in an allogeneic culturing, skipping that step gives you process efficiency, gives you a greater efficiency per footprint, for example. It also eliminates a whole single-use bioreactor of plastic. So it's often process intensification can reduce overall cost, especially over the lifetime of the product, and be a greener manufacturing process. So it's incredibly just a valuable approach that helps in every category with reduced expense.
Yeah, and I guess what that means really for all of us is that the shortest distance between 2 points isn't a straight line; it's skipping steps.
Well, yeah, and reducing, and concurrently reducing many problems. For example, in the automation, you can, even though the per minute process might not be any faster or might be slower than a manual technique, the fact that it can run 24 hours a day, the fact that it doesn't take vacations, the fact that it doesn't introduce contamination, as a person could, overcomes the initial capital expense and the maybe even reduced per minute rate of operation.
Yep. Excellent. So, Bill, in wrapping this up, do you have any other last comments for the audience about process intensification and how it relates to the gaps that you mentioned earlier, looking at the future directions, what the future could hold? Anything else that you'd like to give as a parting thought?
Well, yes. I'd like to reiterate that there are many individual categories of technological innovation that are directly contributing to increased process intensification. We mentioned a few of them. We didn't have time to mention some of them, but they're happening and they're being commercialized. For example, some people don't realize that there are commercial vendors of artificial intelligence capabilities for the pharmaceutical and biopharmaceutical industry that can directly help you to incorporate an AI-based model in process development or manufacturing today. And some people don't realize that. They think about it as this, the way it was 10 years ago is a fanciful potential of the future. No, it's happening today. And I'd like to wrap up on that case study that I introduced that DPS was working on.
Yes.
They ended up with a very nice solution, giving the desired recombinant protein and viral vector manufacturing in parallel, and solved this kind of diverse existing facility, multi-floor, multi-product problem for the client. And this is through, as I mentioned, for AI, for building design and suite design, using automated model building devices. We're able to, I've got the name here. Super Pro and Schedule Pro. We're able to efficiently design this facility to give a higher throughput and a safer manufacturing process.
Excellent. Well, I appreciate that, and thank you for rounding out the case study. I think that's a great result. And also, too, thanks for sharing your part of the 180 cumulative years of expertise that we've had here today.
Well, thank
And
you, Ben. I think you've done a great job. I really appreciate I thought this worked very well. Everyone had a very fluid presentation, and you've done a great job moderating.
thanks, I appreciate that.